Moving larger from peptides, we now consider the structural features of proteins. Proteins, of course, are long polymers of amino acids with molecular weight starting at 20,000 but can go to 200,000 or higher. They're typically going to have over 100 amino acids, up even close to 1,000 amino acids. So the function of proteins in the body, of course, there are four major functions. They're structural, they are catalysts, namely enzymes. They can serve as messengers if they're hormones, or they could be involved in the immune response as antibodies. So in terms of the structure of proteins, which is what we're going to talk about in this lesson, there are four levels of structure. There's primary structure, secondary structure, tertiary structure, and quaternary structure. So the primary structure of a protein refers to how the amino acids are attached to one another using covalent bonds. Okay, so obviously the polypeptide backbone is covalently bonded, and we've already looked at that in detail in the last several lessons. But what we also need to talk about are what is called disulfide bonds, which are covalent bonds formed between cysteine residues that are not next to each other. Okay, so cysteine, of course, is the amino acid with an SH on the side chain, and these two cysteine side chains can be enzymatically oxidized to form an SS bond, and that's called a disulfide bridge. And the process of forming disulfide bonds is referred to as crosslinking, because either two parts of the same chain or even two different chains get linked together covalently, and that helps to fold the protein into its active conformation. Okay, so disulfide bonds are part of primary structure. Then we get to secondary structure. And secondary structure refers to non-covalent interactions between parts of the polypeptide chain that result in characteristic localized structures. So secondary structure is a result usually of hydrogen bonding between the amide NH of one portion of the chain with a suitably located amide CO group of another portion of the chain. Okay, so recall that peptide bonds are planar. So the planar portions are indicated in blue on this diagram, but you can have these blue rectangles twist relative to each other in 3 dimensions, which is how we get folding of the chain and how we get these carbonyl groups and NH groups close enough to each other that they can participate in hydrogen bonding. So the most common secondary structures are what are called the alpha helix and the beta sheet. So I want to look at both of these in turn. So with an alpha helix, the protein starts to coil in the shape of a helix, or in the shape of a corkscrew if you prefer. And it's actually going to be a left-handed helix. So the hydrogen bonds form between a particular carbonyl group and an NH group that's 3.5 amino acids down the chain. And with an alpha helix, as you can see, the R groups, because they're big, point towards the outside of the helix. The beta sheet, on the other hand, is when you get two chains that are essentially linear for at least some distance, and they line up next to each other, such that the carbonyl group of one portion of the chain and the NH of another portion of the chain are close enough to each other that they can interact. Okay, so beta sheets can be thought of as being either parallel or anti-parallel. What a parallel beta sheet means is that both of the chains are going in the same direction. So in the figure on the screen, the N terminus of that portion of the chain is on top and the C terminus is on the bottom. For an anti-parallel beta sheet, they are actually going in opposite directions. So the N terminus of one portion of the chain is on the top and the C terminus is on the bottom. And for the other portion of the chain, the N terminus is on the bottom and the C terminus is on the top. And then you can see how these hydrogen bonds form. So if it's a parallel beta sheet, the NH and the CO are not exactly right next to each other. They're slightly offset. And you have enough room for one R group in the middle, the other one going to the outside. For an anti-parallel beta sheet, they line up perfectly. The NH and the CO line up perfectly. The two chains are just a little bit further apart, and there's actually room for two R groups in the middle. And for the anti-parallel beta sheet, the R groups alternate between being in the middle and being on the outside. Now, they're not completely in the middle because the R groups actually are pointing either in front of or behind the main chain, depending on their stereochemistry, whether they would get a dash or a wedge. Okay, so protein structures are very large, and if we draw out every atom explicitly, we would have an unintelligible mess. So there are some shortcuts that are used to indicate the structure of proteins, and the most common is what's called a ribbon notation. You'll draw side chains as you need them, but if part of the protein is just an alpha helix, you're just going to draw a helix there. And then a beta sheet, a portion that's acting as a beta sheet, just gets a big fat arrow. And the way we indicate the two beta sheets using ribbon notation, if they are anti-parallel, is you have the arrows pointing in opposite directions. If they are parallel, you have the arrows pointing in the same direction. So also notice that when there's just what looks like a rope between a couple of these, what are called domains, that is just random coil peptide. It is neither alpha helix nor beta sheet. So anti-parallel beta sheets need a shorter piece of random coil between the C terminus of one portion of the sheet and the N terminus of the next portion of the sheet. For parallel beta sheets, we need a longer piece of random coil in order to get the N termini to line up next to each other, as is depicted on the screen. Okay, then finally, we get to tertiary structure, which is how alpha helices, beta sheets, and random coils, each of which we call domains, fold together to give the overall three-dimensional protein structure. Okay, so this is a tertiary structure in ribbon diagram of a protein. So you can see there's a couple of alpha helices on the left. Then you have a region of parallel beta sheets. And then you have a region on the right of more alpha helices. And you can see how the different helices line up relative to each other. Okay, and then also notice that any portion that's, again, just linked with a narrow rope is random coil. And notice how long the random coils are between the different pieces of the beta sheet that actually can hydrogen bond to each other. Okay, and this protein is color-coded from a red end to a blue end in the violet end in rainbow colors. So you can see obviously one terminus of the protein is the red end, and then you move through the orange, the yellow, the green, the blue, and then ultimately to the violet. So the general principle for tertiary structure is that if a protein is going to be a soluble protein, soluble in the aqueous environment of a cell or bodily fluids, hydrophilic side chains are going to be exposed on the outside of the protein, and the hydrophobic side chains, made a mistake here, are buried deep. If the protein is a transmembrane protein, so it's sitting inside a cell membrane, the cell membrane is, of course, a hydrophobic environment. So in that case, if it's a transmembrane protein, the hydrophobic regions point outwards in membrane-spanning domains. And then finally, the quaternary structure for proteins that are really, really large and contain more than one polypeptide chain. The quaternary structure is simply how multiple polypeptide chains come together to give an intact functional protein. Okay, so depicted here is the structure of hemoglobin, which is a protein that contains 4 different subunits, we call it, 4 different polypeptide chains. You can also see the porphyrin units indicated in more of a ball and stick drawing rather than the ribbon drawing. And this is shown so that each of the 4 subunits, the 4 independent polypeptide chains that each have an N-terminus and a C-terminus, are a different color. Okay, so you have the red chain, the yellow chain, the green chain, and the blue chain. Okay, so that shows now how you can get a huge protein, and it all comes together using the principles of organic chemistry that we've learned over the course of this entire semester. Okay, so the coupling reactions of amino acids to form peptide bonds, hydrogen bonding to form the secondary structures, and then hydrophobic versus hydrophilic properties govern how the tertiary structure and the quaternary structure come together. And so building up from atoms of carbon, hydrogen, nitrogen, oxygen, and a few important sulfurs in there, you can make these massive machines that can carry out most of the major life functions just using the simple organic reactions that we have learned about. A logical extension of this then would be to look at how enzymes help to catalyze the organic reactions that take place in the body, and then that would serve as the bridge between organic chemistry and biochemistry.